Hello. What scientific topics deserve the reverse methodology approach? That's what I cover today. I'm Dr. Mark Williamson for Transcend. Let's get started. So, what is reverse methodology? Broadly speaking, it's when you switch a research methodology from one direction to another. So, a core field would be something like translational research, and a core example would be epidemiology. What do I mean by that? Well, let's get the big picture here, and then I'll give the epidemiology example. So, a pretty traditional research approach, especially in translational research, is you start in the laboratory, so a controlled space, controlled experiment, where you can sort of keep everything neat and tidy. And then, if you find promising results, you might try to translate it into a clinical trial, some sort of intervention, and that would take place in the real world, which is sort of uncontrolled space. It's messier. So you really want to have your ducks in a row from the lab before you go into the real world. However, a reverse methodology actually doesn't do that. It sort of reverses that. You'll start in the real world, maybe from some observation, from some correlation, that is promising, and then you go back to the lab to sort of experimentally confirm those observations, those hunches, etc. And so this can be really powerful. And the example I'm talking about in epidemiology is what might be called trench to bench. So traditional translational paradigm, something like bench to bedside, you go from the lab to hopefully improving patients' lives, so, the actual bedside for patients. Or you could think of bench to trench, that's more epidemiological. So you go from the laboratory to the population, so communicating epidemiological outcomes to the public. But the reverse methodology would be like the trench to bench. So maybe you start with epidemiological insights, so maybe a potential risk factor, environmental risk factor in a disease, and then you go back to the lab to try to confirm that using like a mouse model, cell culture model, etc. And so this can be very underutilized and but very powerful. And so it's important to have the idea of reverse methodology as a potential tool in your scientific explorations. And so that's the rest of this topic here. This talk, this video is sort of brainstorming more topics that this would apply to. Not that it doesn't already apply to this, but maybe helps juice your thinking into more ways a reverse methodology could be used. So my alpha list is maybe the most straightforward ones. Then I have a beta list where I get a little more wonky. So first one on the alpha list would be clinical observations. Pretty straightforward. This traditional approach would be, you're doing a basic lab, basic scientist has some sort of insights into, say, like drug treatment and tries to go apply it after promising results, maybe in a clinic. Uh, the reverse methodology would be, say, maybe starting with clinical observations, maybe a clinician in his practice notice maybe a potential correlation between like adverse effects in, say, a drug and a certain subpopulation. And so then maybe he would go from there to the lab and maybe test that in a more controlled environment to see if it holds up. Another standard reverse approach might be environmental exposure. Again, that's going into that epidemiological, but it doesn't necessarily just need to be epidemiology, but something like that. Rather than starting in the lab with testing things and then maybe going out into the environment, maybe you start with environmental observations like, oh, we see this outcome and this environmental exposure, whether it's plant, whether it's pollution, whether it's something like that, and then taking it back to the lab. Three would be chemical synthesis. I think this one might work, instead of like a priori thinking of like chemicals you want to synthesize and try to synthesize in the lab for some later use, some later application. Maybe you would start with examining chemicals already being synthesized or already able to be synthesized, and then going back and seeing if they have uses or can be reverse synthesized or something like that. A similar relationship would be bioprospecting. That also happens. So instead of maybe trying to design from scratch biological organism or some sort of protein product or something from sort of biological cultures and then apply it down the line, maybe you would start by looking at the environment, like going and getting a bunch of metagenomic samples from like deep sea mud, and then metagenomic sequencing or proteomics to like look at what these things are actually producing, and then selecting interesting things from there to bring back to formal lab experiments. This third one, stay with me here, but might work here is what I call like sort of literature review or idea or hypothesis review. So rather than in a given field necessarily having to sort of generate your own hypothesis from scratch and then trying to, write a paper for them, after testing them, you could imagine a reverse methodology as saying like, what interesting unanswered questions or unanswered hypotheses already exist in the literature, look at them, and then maybe go back and test them. I think that'd be really fascinating for people to do more of that. All right, let's get to the beta list where it gets a little more wonky, a little more conjectural. So frugal science. So I already did actually a science spotlight video on what frugal science is. You can certainly go check that out if you want. But but generally speaking, rather than sort of having an engineering or sort of science solution and trying to generate from scratch, to have some sort of application, what if you already take common applications um, to reverse engineer and then go back to the lab to use as a low cost alternative to whatever methodology, whatever equipment you need. Another one would be mental health. Again, this is kind of getting out there, certainly outside my expertise, but rather than saying, well, we have these mental health things and we have these potential interventions and we're going to test them in the lab or test them and before trying to apply them, maybe the reverse methodology would be helpful. Like look at the population already and like, well, what solutions, like what sort of things that people do to treat their mental health or sort of address their mental health already have success. And then try to, look at that more rigorously rather than trying to like come up with solutions that apparently clearly aren't working because, mental health, despite there being more sort of mental health resources than ever, mental health rates, are not improving. They're getting worse. Anyways, so on more of the biological, the more like physical side of things, you could think of disease remission instead of saying like, well, let's try all these experiments to see if we can get disease to go into remission. What if we looked at the reverse, looking at cases in the population of people who've had sort of spontaneous or unexplained disease remission and see if you can figure out why and then maybe test that why in the lab, whether that's cancer, autoimmune disorders, etc. Fourth one is phytoremediation. So again, that's the idea of remediating places like, especially like soils with plants, or just remediation in general. So rather than say, okay, we want to test certain compounds, certain plants out, see how good they are, maybe you would reverse it. Go out to places that have been disturbed that need remediation, see what plants or organisms, even algae, even like soil bacteria, microbiome that are already remediation, see if you can reverse engineer that. And finally, I sort of piggybacking off that is space biology. Specifically, like thinking about, again, I did a video about simulated lunar regolith with plant studies. So there's a lot of work on like trying to modify lunar regolith so you can grow crops in it. But and I think a really great reverse methodology approach is rather than saying, well, I have a bunch of these potential plants I'm going to test and see if they work, and if they work, kind of move on here. Maybe you go back to say, if I'm just going to take regolith, see what grows, so the reverse methodology, and then take the ones that grew, from maybe a wild seed population or whatever, just keep it a natural experiment, and then take those back to the lab and then see if you can develop that further. Anyways, so that's a little bit about reverse methodology, maybe some avenues that could grow in that. Certainly, I'm sure there are more. You can leave that in the comment below. Hopefully, this was helpful. And with that, I will see you next time.